Okay, let's look at protein. Protein is, of all the macromolecule, protein is the most diverse macromolecule. And we tend to think of protein typically of when we consume proteins as far as what muscles are made of, right? But proteins play so much more of a function than just muscles. So protein is, as I mentioned, the most diverse in terms of its structure and its function. And so proteins have quite a diverse set of functions, right? Proteins can actually play a role with regards to structural support. And so, and you can see the structural support in cells, organs, and tissues. They play a big role in structure. We can see proteins are immersed. Even remember how we talked about the cell membrane when we looked at carbohydrates and how they made phospholipid, but we can actually have a lot of proteins in there as well, and they kind of function as far as structure, as far as communicating. Our muscles are made of protein, so when we think of our muscles are made of proteins as well. And so when we are actually contracting and relaxing, that's actually our muscle contracting, and that's the proteins that's functioning there. Proteins make up our antibody. So when you are challenged with a virus infecting, and your body is able to after it's able to defeat the virus, it can form these memory cells that are antibodies that it can then use later on to kind of mark the next time that you're attacked by like a virus. So they can function in immunity, they can function in enzymes. So remember how before I talked to you about how there's an enzyme that can convert with regards to high fructose corn syrup, the glucose into fructose. That's an example of an enzyme. So enzymes are basically proteins that kind of help catalyze certain reactions that might not otherwise be able to react and fully complete if it wasn't for certain enzymes. Enzymes can also function as hormone. So insulin is an example of that. They can also function with regards to transport. So hemoglobin is actually in your red blood cell that kind of carries oxygen to all the different parts of your body. So protein plays a big function with regards to transport. And then if your body can also use, as I mentioned, your body can use protein and lipids as energy source as well. It's not its first choice, but if you no longer have a carbohydrate storage available, all your carbohydrate depleted, your body can actually turn to protein next as far as deriving energy from it. So the next time that you eat these different protein food components, just think about like how much they're allowing your body to do, everything from structural to actually muscle contractions. So even when you're standing up rigidly and walking, even just standing still, your muscles contracting. And if it wasn't for protein, you wouldn't be able to have that rigid stand. So protein just plays multiple functions in your body and has quite a wide range. So we should like learn to appreciate protein a little more because it plays such a big role in our body system. So what are proteins made of? So a protein is actually made of, it can be made, and we'll see this when we actually look at the structure, it can be made of multiple polypeptide. And polypeptide is the polymer of a protein structure, and these polypeptides are made of monomers called amino acid. So this is a monomer. A monomer of protein is an amino acid. And we actually have multiple different amino acids. There are 20 different amino acids, and they vary based on this R group right here. So all amino acids have an amino group and a carboxyl group, but they also have this R group, and this R group can vary quite a bit. And so you can see over here that these are different R groups that are in the blue, right here, right here, right here. And so these R groups can be polar, as we see here. The reason why I know it's a polar is because of that negative charge that's associated with that oxygen. It can be non-polar, right? So this right here is all just carbon and hydrogen, and we've seen before that if it's just carbon and hydrogen, it tends to be non-polar. So it can be polar, it can be non-polar, and because of those characteristics, every R group has a specific characteristic to it, and it's what makes it so that proteins is so diverse, because depending on what combination of amino acids you combine together will change the structure and the function of the protein. Okay, so again, amino acids, they're linked together by peptide bond. That's why it's called a polypeptide. But first, let's try and look at two different amino acids. So here is amino acid, and we're just going to assume that this is R group number 1, and this is R group number 2, and they kind of have slightly different, they just have different R groups to it, right? But all amino acids have, as we mentioned before, an amino group and a carboxyl group. So this carboxyl group and this amino group are going to link together through what type of reaction? I hope you guys said a dehydration synthesis reaction, as we're moving down this way, right? And as we're moving down this way and we combine this hydrogen and this hydroxyl, we're going to form water. And in the process, we're going to link these two together, and we're going to link them by something called a peptide bond. So this is a peptide bond right here. This carbon right here from this carboxyl group and this nitrogen right here from this amino group are going to combine together and form a peptide bond. Multiple peptide bond results in a polypeptide. So right, so a polymer of protein is a polypeptide, and it's because these individual monomers of protein, also known as amino acid, will link together via a peptide bond. Multiple peptide bonds leads to a polypeptide. And this is a covalent bond, so that means that this carbon and this nitrogen are sharing their electrons equally. And so we can have a polypeptide that's multiple amino acids long together. And keep in mind that each one of these amino acids, so we'll put 1 here and 2 here, are going to have different R groups again. So just to iterate, what type of reaction forms a polypeptide? So what do we have happening here? We have the carboxyl and the amino group reacting together. They're going to, the carboxyl and hydrogen is going to end up forming water. And between the carboxyl and the amino group, we're going to have a peptide. And so this right here is also known as a dehydration. I'm just going to, actually, let me type it out. So above here, we have a dehydration synthesis, right? So the reaction that forms the peptide, polypeptide, is a dehydration synthesis. Well, what if we want to break them up? Now, remember that when we are talking about polymer or monomer, we can synthesize polymer by adding multiple monomers together, but we can also break up a polypeptide back to its monomer. So this would be a hydrolysis reaction. If we want to actually take this and we add water to it, we can have the reaction pushed this way and break this polypeptide back to its amino acid monomers. Let's move this over. Okay, so we talked about how proteins can have such a wide variety of structure and function. So why are they so different? Why are they so diverse? Well, they're diverse because of these different R groups that we had mentioned before. There's multiple R groups. Well, these R groups have different characteristics to them and can lead to different type of folding, right? So the primary structure is just the amino acid sequence itself. So you have your amino group at this very end right here. And then we don't see it here, but over here, we're going to have our carboxyl group. So I'm just going to put C for carboxyl. And the carboxyl, actually, let's just kind of draw it out a little bit so we remember. The carboxyl group is going to have a double bond with an oxygen. And it's also going to have a hydroxyl group. So we're just not seeing the whole thing because it's really long. So a primary structure, a lot of times we'll also see it being marked as 1 degree right there, has a bunch of amino acids. So here we're labeling them based on their name. So this is a valine, this is adenine, this is the alanine, this is a lysine. So these are all different amino acids, and they all vary based on their R group. So the R groups are just different. And so this is the primary structure. It's just the actual sequence of it. We move on from the primary structures to the secondary and the tertiary structure. So what are these? Well, the secondary structure is when the polypeptide changes full and they form hydrogen bond. Remember, hydrogen bonds is between hydrogen and an electronegative group right here. So you can see here, this hydrogen wants to be near this oxygen because this oxygen is electronegative. So we're just forming these, this hydrogen here is interacting with this oxygen. So we're forming these different hydrogen bond. And when we form this hydrogen bond, it results in the polypeptide just kind of folding, right? So here you have it folding here. So they're not, remember, hydrogen bonds aren't actually, they're not actually sharing the electrons, right? And they're not transferring electrons either. So they're not actually holding hands like this, where they're sharing the electrons, where they're each one pulling an electron. They're more like just really attracted to one another. So they're kind of near each other, and they're really attracted to another. And that kind of makes it so that they will fold and change their shape, but they're not actually linking together. They're just kind of really close to one another. So this results in these weak interaction as they interact with one another. Your tertiary structure is when you have even more folding. So now these foldings together from just the secondary structure is just the alpha helix itself, or the beta sheet right here. And the tertiary structure is when you actually have it forming a more compact. So this is all the folding. So this includes the alpha helix, which we see here, but it also includes the beta sheet folding that we see here. So this is a combination of all the different type of foldings that you're actually seeing happening in a polypeptide. And so now we can form a functional protein. So this right here can be one polypeptide that forms a functional protein, but oftentimes we have more than one polypeptide. So oftentimes in a final protein, we have 2 or more polypeptide that are associated. So hemoglobin is an example of that. So what we mean by that is here, this can be a fully functional protein, just one polypeptide. And this would be the tertiary structure right here. But we can also have multiple polypeptide actually folding, each individually folding, but through hydrogen bond, through interaction, they can actually form this quaternary structure. And so our final protein can actually be multiple polypeptide interacting together. And so this is what makes proteins so diverse, is because of the different ways that they can fold, the different amino acids to begin with, the different ways that they can fold, and the fact that you can have multiple polypeptides folding together to form a functioning protein. And so one of the things that we should keep in mind is that for proteins to be able to keep its structure, to be able to keep its function, I should say, for it to keep its function, so let's say for hemoglobin, for example, for hemoglobin to function properly, it has to have a specific structure to it. And so we can actually disrupt that 3D structure. Because remember, a lot of these 3D structures is based together by these hydrogen bonds as well as other weak bonds, which we didn't really go over yet, but there are other weak bonds that are allowing the protein to interact together. So we're going to focus on hydrogen bond because that's the one that we've looked at already. So, but we have also van der Waals bonds and different type of cysteine bond and stuff like that. That's also forming different linkage or different interaction together. So because hydrogen bonds, we saw with water, can be broken, right? As we said, they're not linking together. It's just kind of near each other. So when you have these bonds that are near each other, we can actually break that bond. And we can break it through changing our pH, by changing the temperature. And when we actually break it so that the bond that was originally here, like we have this hydrogen bond, and we actually break it so they're not linked together, we can actually destroy that structure, right? Because if this is what's keeping the structure together, it's these different bonds, like forming the alpha helix. But if you break that alpha helix, it no longer has that same folding. So it's what we call denaturing. And so when we denature, we're actually breaking those bonds. And by breaking those different bonds, we actually can influence the shape of the protein and disrupt the function. So one way that we can kind of think about this is we know that egg white has a lot of protein, right? So egg whites can actually provide support for the growing embryo that's growing the egg yolk when an egg is fertilized and developing. Egg white also has a lot of nutrient benefits and so for the growing embryo. So egg white plays a pretty big function for this growing embryo. But when we crack an egg white, right, so it has a specific structure and function to it, right? When we crack the egg white and we add heat to it, what happens? Well, when we add heat to it, it actually changes its structure. And we can't take this structure and take it back to the way the egg white looked before we added the heat. So this is an example of denaturing, is when we actually take that egg white, we add heat to it, we're actually denaturing the egg white right here. So we're kind of causing it to unfold, and that is an example of the denaturing. Oops. Okay, so that's my last slide for proteins. So remember again that proteins have so many different function and structure. Recognize that the structure and function of protein is based on what? It's based on how it folds, right? So the structure of the protein determines its function. So what determines the structure of the protein? Well, the structure of protein is determined by how it folds. Well, what determines how it folds? The amino acid. The amino acid that makes up the primary structure of the polypeptide is what makes it so that different monomers of the polypeptide with the different R groups will interact with each other to form hydrogen bonds, to form van der Waals bond, to just kind of form different linkage, right? And so that's what defines the structure and hence the function of proteins.